Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Douglas Storace, a neuroscientist and assistant professor at Florida State University. Doug is also a 2022 Toffler Scholar. We will be discussing his work in understanding the olfactory bulb, that part of the brain region responsible for processing smell, and its implications to the overall function of the brain and our bodies. So let's dive in. Welcome, Doug.
Thank you, Deborah. Thank you for having me. And everyone says this, but I do really want to express my gratitude to the Toffler Foundation for that funding. Hopefully I'll be able to talk a little bit about that project, but thanks for inviting me.
Yeah, I would love to hear more about the project, because I think we talked, my goodness, maybe 18 months ago, and I'm sure there's been a lot of really interesting progress. So I'm looking forward to hearing about that. I just kind of want to start with what sparked your interest in neuroscience? Why this field?
Yeah, it's, would you believe I never ever thought about the brain before I took a course in sensation and perception with who ended up becoming my PhD advisor. I don't think I ever thought about it at all. And I was interested in computers. I kind of enjoyed programming. I wasn't particularly great at it. I liked the idea of it. But then I took this course, and I remember when we covered the visual system, and I was like, it's really just that is very interesting. It's very modular. It's very structured. And it was organized, kind of in a computational way that just made sense. And I was kind of going through a music obsessed phase. And that teacher was actually she studied the auditory system. And early on, maybe, I was interested in playing music. And I had some kind of I would call them superficial or maybe kind of almost silly questions about the brain and sound. But it definitely got me interested in learning more about the neuroscience of sound. And I ended up working in Heather Reed's lab, who ended up being my PhD advisor as an undergrad during my senior year. And I was very fortunate that she accepted me into her lab for my PhD. So it was really the connection of how to think about the brain as a computer, which maybe seems super obvious to everyone now, but maybe it's just not something I had thought about. And then at the moment, strong interest in music, and happened to be my teacher was studying sound. I was like, okay, this seems really up my alley.
So that's interesting. So you said when you didn't really think about the brain when it came to any of these sensory kind of inputs, but you said it made sense because it was very structured. Can you just kind of expand on structured how? How did that impact your thinking?
Literally, okay. It's a little bit some parts of your eye, there's a spatial mapping from your eye to different parts of your brain. So this part of your eye goes to this part of your brain, and this part of your eye, I'm mixing them up, go to a different, no one can see me, I'm sorry. There is a literal spatial mapping from your eye to different parts of your brain. And through the connections from in your eye to these areas, the neurons connect together and they perform different functions. And so that is, I literally mean the physical structure made me say okay, that's really like how computers are working. And so it was very appealing to me and intuitive.
So those structures then is not just the optical, the eyes, but also the hearing and the smelling. And does taste also have that structural kind of component to the brain?
That's a very interesting question. And I am a lump. One of my advisors told me you can either be a lumper or a splitter. Dr. Harvey Swadlow, I'm kind of a lumper of ideas. I tend to try to find things that are similar. So yeah, this topography is present in many sensory systems. Taste, it's a little bit controversial of whether it's similar, but I think there are commonalities across the different sensory systems that I hope can teach guiding principles that can be useful in understanding the brain. Maybe you have totally different inputs, right? Sound is different than light, is different than smell, is different than pressure. And so the brain has to detect them differently, but that doesn't mean that the circuits that are used to inform about that information and our perception, like perception being like our experience of everything, has to be different. There might be common patterns that are repeatable.
And repeatable across people or across the experience of a person?
How I meant it was across brains, within the brain, let's say, and maybe this is getting a little bit far ahead, but in the auditory system, you worry about masking. Like, how do you detect sounds in the presence of noise? Right? How do we detect things that we care about? And there's a famous example that everyone always talks about called the cocktail party effect, which is that your brain can tune to some stimuli and to some sounds, try to minimize the jargon, and somehow ignore stuff. Right? You kind of know what to pay attention to, and I'm going to introduce that concept as segmentation, background segmentation. And you have the same problem in smell, which let's say I have a picture that I love of a mouse trying to find some cheese on a paper I wrote a couple of years ago, and I put it in all my grant submissions. And the mouse trying to find food, but there's lots of stuff in the environment, and your brain has to ignore that stuff and pay attention to the thing that matters. And you might think that maybe there are common motifs of how the neurons are organized to perform those functions. Right? It's the same thing that needs to be done. Segmentation, adaptive function, basically. So that's kind of what I meant is, maybe you address the similar question in a different sensory system and you see that, yeah, there is a different organization, but the underlying principles are quite similar. Maybe, right? That's how I'm, as a lumper of ideas, that's one of my guiding philosophies.
Super interesting. So what is the focus of your lab, and what is the key kind of questions or hypotheses that you are trying to test? Because the way I understand just what you said is there is a brain is kind of the central control point, but you're looking at all the sensor inputs and how they relate to the brain. So you're kind of taking maybe that translational look from the outside environment, and then how does that work, on the brain or with the brain? Am I understanding that correctly?
Yeah, we have a couple of different big picture projects. They're each interesting in a different way, and I think I can explain them in a non-too jargony way. So you tell me if I've gone off the deep end here. But the first one is still related to what the Toffler Foundation, and I think I need to describe a framework of what we study. We study the olfactory bulb. This is a brain area at the intersection of the outside world and the first entry into the brain. And it's a little bit analogous to the retina of the olfactory system. Right? And now, like, light comes in and the cells in your eye transmit those signals into your brain, the highest part of your brain. So is the olfactory bulb. But it's this peripheral brain area. And a framework that I think makes it a little bit more interesting to think about is like sensory information comes into your nose, and your nose is telling the olfactory bulb something. And for a long time, people thought of it as comes into the brain and then it goes somewhere else, and then some processing occurs, something, the brain is doing something to that information. But I think the last maybe 2 or 3 decades, people are appreciating that there are loops, and I'm going to call them loops, which is that the nose is telling the olfactory bulbs things, but the rest of the brain loops right back into the olfactory bulb and is telling the olfactory bulb things as well. And the reason that's interesting is because it means the brain can shape, transform, modulate, which jargony word you want to choose, the actual sensory input, right? What your brain is even getting about smell can be automatically shaped by the rest of the brain. And so that's kind of an interesting idea. Like lots of different brain areas feed into the olfactory bulb structure. And so it's not a new observation, but that this phenomena exists. But what was really an interesting preliminary data that I submitted to the Toffler Foundation, which is my graduate student, Major Key, who will be on the job market in the next year, looking hopefully for scientific positions, but who knows. She found that a very interesting brain area fed into the olfactory bulb. It's called the hypothalamus, and this brain area is very interesting because it's like kind of in the center of the brain, and it's involved in many key behavioral functions. Like in my textbook that I teach from, it has examples from like the 50s. People would stimulate the hypothalamus, animals would start eating, or they would, stop eating, if you stimulate, or they'll fall asleep, or they'll wake up, and so brain area involved in sleep, feeding, mating, drinking, circadian rhythms, all these different functions. Very, very interesting observation that the olfactory bulb is getting input from a brain area involved in state. She also found that, I'm going to define one more concept. I'd say a major conceptual revolution, and I don't think that's an overstatement, of the last 20 years or so was the idea of different cell types. And what does that mean, right? If you look in a brain, the cells all look roughly the same. For I'll give an example of how difficult things were. My PhD advisor, not too long ago, but they would try to differentiate between different cells based on their shape, right? They would look at their shape and they say, this one's like starry shaped, and this one is round, and this one is large, and this one is slightly smaller. It's the same idea, but now we're doing better because we can define what makes two cells different based on their gene expression patterns. Like what makes a dopamine cell different than a serotonin cell, different than a glutamate cell, right? There's genetic features that allow us to more precisely study the brain. Now instead of saying we're just sticking the electrode here, or we're looking in this big area, we're studying a more precisely defined collection of cells. Okay, I just think that framework is important for appreciating Major's discovery and what we were doing. But going back to the hypothalamus, she discovered that a group of neurons in the hypothalamus go to the olfactory bulb that express a very interesting chemical. I'm going to call it a chemical because it's my least jargoning way. It's actually a neuropeptide, whatever, it's jargon. It's a chemical that the brain uses to signal from neuron to neuron. And there's lots of these. But the one that I'm going to tell you about is very interesting for several reasons. It's called orexin. It's also called hypocretin. Just a little bit of background. In 1998, 2 groups simultaneously discovered it. One group found that this chemical made animals eat when putting into their brain, like if they just injected it into their brain. So they called it orexin because it's related to appetite. The other group found that it was strongly related to sleep. And turns out narcolepsy is a disease that's related to dysfunction of the orexin system. The two groups gave it different names, though. So one group called it orexin, the other group called it hypocretin. A little bit confusing. You don't want to use the wrong word because some people, whoever, probably they like their name better. So if you're playing it safe, you're going to call it orexin hypocretin neurons. Anyway, very interesting. Another interesting thing about these neurons is that they're in a human brain. Maybe there's billions of neurons, right? Many. Who knows how precise that count is? But a lot. In the whole human brain, the estimates are that there's about 10,000 of these neurons that make this chemical, which is very interesting. Tiny number. And in rodents, it's about 3,000. So in a whole brain, that's really not that many cells. And they're all clustered together in the hypothalamus. So it's kind of this interesting population of cells that can broadcast a particular signaling molecule to the rest of the brain. And this molecule, orexin or hypocretin, over now the last 26 years since it was discovered, elevates metabolism, definitely involved in sleep. There actually, I was looking up, there's people have knocking it out can cause obesity in animals. It's definitely related. It's related to all these state functions. And there's actually been clinical trials to develop agonists and antagonists of orexins, which are drugs that bind or block the system for many, many different kinds of diseases, certainly narcolepsy, but also mood disorder, depressive disorders, insomnia. If it's narcolepsy, just do the opposite for insomnia, and even drug addiction. So very interesting, right? It's like this small cluster of cells deep in the brain that are possibly involved in all these functions. We study the olfactory bulb. So we mostly thought of it as an interesting, plausible mechanism by which a sensory area could be controlled or at least shaped by your internal state. I'm going to say some speculative things now because the literature on this is a little bit messy in olfaction, but I think it's probably true that our sensory sensitivity to things changes, like if you're hungry or not hungry, or if you're tired or not tired. It's difficult to do these experiments, and there are papers claiming actually in both directions of these things. But we were really excited to think of this pathway as a way to, like, if you were hungry, maybe the brain wants to enhance your sensitivity to some smells or make it easier to detect some smells in odorous or noisy backgrounds. Or maybe if you're sleeping, maybe it wants to downregulate it so you're, less, right? Just that's the very kind of simple elevator pitch explanation for how we were thinking of it conceptually. Okay, that is one big picture project of what we're studying. I didn't tell you how we're studying or anything like that, but just the concept.
Yeah, so just for clarification, what your hypothesis then is that the brain is doing something through the hippocampus, right?
Hypothalamus.
The hypothalamus with its relationship to the olfactory bulb to basically influence behavior. Go eat, go sleep, shut your eyes, focus on this sound. You're in danger, shut down these things and only focus on these things, whatever those things are. And that is a new concept from what I hear, is that how these relationships have been working with the brain and with these other sensory inputs, especially as they relate to the outside environment, which we live in.
Yeah. Like these orexin neurons. So in the hypothalamus, I'll just give you I'll give you two interesting examples. There is a part of the hypothalamus that contains neurons that can detect a chemical that's released by your fat cells that is a cue for how hungry you are, called leptin. So there's literally neurons in this brain area that can sense the chemistry of your internal state, and it's being transmitted directly into your brain. Another example is that the retina, which is detecting light, sends signals directly to the hypothalamus into a suprachiasmatic nucleus, which is thought to be important in circadian rhythms. So this brain area is really getting, like, it's at this interface. It gets some external information, but it's also regulating internal state through sensing of body chemistry. But you're also correct that, I mean, it's really, I think, in even the last 15 years, the appreciation that there's this relationship between the body and the brain is exploded. There are now many groups studying the nerve innervation into the brain. And the vagus nerve is a big, I don't study that, but that's a big deal now. And some of it is because there are groups that figured out how to selectively study the vagus innervation from different parts of your body, right? Maybe the vagus is innervating, connecting your brain to your gut, or it's connecting your brain to your lungs or your heart. And these are different pathways. And so now there are these tools going back to cell specificity. You have tools to better dissect out those pathways and really appreciate that there is this literal communication pathway that connect your brain and your body.
Yeah, that's fascinating.
It's very interesting conceptually.
Yeah. So what's the second major project that you're doing in your lab?
Yeah, the second project is actually related a little bit to what I told you about the idea of background segmentation. It's currently an NIH-funded project, and it's being led by two people in my lab, a postdoctoral fellow. I mentioned majors, so I'm not going to not mention the other people, but Li-Min Liang, who's a postdoctoral fellow in my lab, and a graduate student named Priya. And it's really all about how the brain, how we maintain perceptual stability in different environments, right? How in noisy places can we maintain focus on something? Like, how does the brain, how is it if you smell something that's very faint or very strong, you know it's the same thing? How is it that your brain segments, let's say, some important food smell from a environment that can contain many, many odor molecules? There's a group that has studied Drosophila. These organisms are so good at detecting vinegar. They are amazing at it. They can find it from like 100 yards away. I mean, when you think about how small a fruit fly is and how many competing odor molecules must be present that could detract or distract the fruit fly from finding its goal, it's very interesting concept. And the project is also interesting because we think some computations related to this problem are happening in the olfactory bulb. And we proposed a few different mechanistic approaches to try to find how it could be happening. And going back to me being a lumper of ideas, the hope is to kind of find common principles of mechanism and organization that might be similar in other brain areas. Like maybe the auditory system ultimately uses a similar computational process as the olfactory system to achieve those goals. And then the third project is really crazy and it's very, very interesting. It's being spearheaded by a postdoctoral fellow named Evan Lloyd, who essentially he brought the project to me. He is studying smell in a fish. And this is a very interesting fish. It's a morph of a Mexican tetra, which is a kind of a common fish. But some groups in the last maybe 100 years, maybe like a couple of decades ago, found in some caves in Mexico that they found descendants of the Mexican tetra that have been living in these caves for perhaps, it's not clear, but perhaps maybe 100,000 years. And they evolved and thrived in this unusual, lightless, and weird environment. You could imagine this did weird stuff to their brains. Notably, they're called blind cavefish because their eyes deteriorate as they develop. There are also many other strange properties in these fish. They have weird metabolic phenotypes, like properties. They age differently. And it's fascinating because they have evolved, but we have the original species that they descended from still exist. So it's a very interesting comparative approach. You have regular surface fish, and then you have their weird evolved blind morphs that they were identical 100,000 years ago. And so you have this fascinating way to study how changing a dramatic change to their brain, in this case vision, may have altered their sense of smell. And it's now we're getting to speculation and controversy. There's reason to think they might have some enhanced senses in other areas. Touch, possibly. They're swimming around, so they need to be really sensitive to small vibrations. But Evan thinks that they may have evolved enhanced smell. So there's a couple of groups now studying this, and he wanted to use some of the optical techniques that our lab uses to study their brains. So that is like a completely I was like, man, that's too interesting to not try to get going in the lab. So that's a third kind of off the wall project that we have going on right now.
And is that, somewhere along the way, does that then relate to the human brain in trying to understand, if we lose our eyesight, our sense of hearing may, increase, and, why, right? As you were talking, I was thinking, about just in stressful situations when you're tired and maybe, whatever it may be, hungry, all of a sudden you find yourself in a crowded room and you're having to focus. It's very hard. And then what I'm gathering from you is we really don't understand why. We don't understand kind of this behavioral pieces that we just all take for granted that says, when I'm hungry, I better not be doing really detailed work because I won't be able to focus. Or if I haven't had good night's sleep in a few days, I better not try to tackle something that's really complex or emotional with somebody because, I may not be at my best. Am I understanding that correctly? That we're trying to understand this very complex kind of relationship with what's going on inside and why we react the way we react?
Yeah, I think that's a great way to explain the kinds of things we're interested in a very big picture way. I think it's a really hard problem. The scale of understanding and relating our brains to our perception and experiences is just so difficult because part of it is the things inside are very small, and that makes it hard to measure them. There's small electrical signals that are hard to separate out. And then there's many, many cells, and there are many different types, and they all do different things, and they're changing. And there are places in the brain that regrow new cells. So it's really a moving target. Really, really tricky. Very interesting problem. I mean, I know the Toffler Foundation is more of a disease. They've traditionally funded many disease models, and I'm a systems neuroscientist with more psychology background. So I really do think about it in more of the terms as you described. Those are the kinds of things I'm interested in, is like what makes us human and to be able to perceive and understand things.
Yeah, I think the expanding our view of what the brain is and what does it do and how does it drive, so many different things is really important, because if we only study the disease, we may not know what causes the disease. And at some point, your research is giving a bigger picture of, what's happening around us, the behavior of around us, and then is any of this causing the diseases that affect the brain?
If we don't understand function, it's going to make it very hard to understand dysfunction. I think I'm taking this quote from one of my colleagues,
Yeah.
Adam Dewan, but I think it's a great point to make because and so I'm okay with that. I philosophically feel that that's true.
Yeah, I was fascinated in a previous conversation that we had about how you were explaining, and I may get this wrong, so correct me if I'm wrong, but we don't really understand when we're baking something and we smell this wonderful, fresh bread or whatever it may be, we don't really understand it. It comes through our nose, it hits our brain, and then our bellies growl, like what is that reaction? I always wonder why, I go and I lift weights, and if I'm lifting very heavy, why at almost exactly 37, 38 minutes into the lift, I'm hungry, even if I ate a big breakfast right beforehand. And these are the things that you're trying to understand if I'm understanding exactly what your focus is.
Yeah, I'm comfortable with your general assessment. I think that's an intuitive and reasonable way to talk about it.
Which is something we take for granted. We take for granted that, we're walking around and these things are happening to us and we don't know.
I lived my life without ever thinking about the brain. You're like, wow, this is a lot that's happening without us really thinking about it. It just is all working out okay. It's actually a miracle that it's working most of the time when you think how many things have to be operating correctly.
Yeah, it really is. So, that's pretty amazing. What are some of the kind of the tools? Because you said these are the projects you're focused on.
Yes, the big
but you didn't
picture.
want to tell us how you were doing it. So, what are some of the tools or the technologies that you're using inside the brain to really understand this function?
Yeah, this is like some pretty interesting stuff. So the reason I switched from hearing or sound, so I did my PhD in auditory neuroscience, and then I was actually looking at positions in labs studying hearing, but then who ended up becoming my postdoctoral mentor gave a seminar, and he studied the olfactory bulb. And he gave a seminar, and there were two things that I remember. One is that he was able to identify different cell types, which was this concept. Discussed this framework before, which was really, really interesting to me because it's kind of a mess. Like you just like would stick an electrode in a brain area and record, and you didn't really know where you were recording, and you were kind of like without any kind of real. It was just you felt like a little bit blind, like to distinguish between all of the millions and billions of neurons. And so he was at the time using tools to differentiate them. And he was also using imaging techniques to differentiate between them. And that gives me a chance to tell you a little bit about some of the imaging tools that are really interesting. And I actually love talking about some of these tools. A lot of neuroscience involves a tool called fluorescent proteins. And these people may have experienced these. If you've ever been to an aquarium, they might have fluorescent lights and you can see like things glow. And I'm not going to talk in much detail about what fluorescence is unless you ask me, because it can get into the weeds a little bit, but it's basically a tool that lets us see some things and not others or enhance contrasts. It's like a marker we can use. Okay, let me tell you about green fluorescent protein. It was identified maybe I think in like the 50s or 60s as being in jellyfish. And I'm not going to even try to pronounce the name of these jellyfish, but the people identified that they glow. And long story short, they chopped up a lot of jellyfish and figured out that there was a protein, a gene encoding a protein that would make them glow. Ultimately, it's called green fluorescent protein. And the amazing thing is that you can take this gene and put it in many, many organisms, even though it's in a jellyfish. You can put it in a mouse brain, and its brain will start, the neurons or the cells that make that gene or that have that gene will start to make the protein, and it closes. Put it in a worm, put it in a fly. Evolution, right? It makes sense. It's the same machinery, but it's still amazing. Many groups realized how useful this tool could be, because now you could put a gene that makes a cell visible in cells. So now we can actually see them because they're fluorescing or glowing. I'm going to use it in a general way. And so there was several decades of making it better, which is a really interesting field of neuroscience research, which I would maybe put into the category of bioengineering, where they're literally figuring out how to make the protein brighter, right? It comes out of the jellyfish, and the jelly is working for the jellyfish, but maybe it needs to be like 3 orders of magnitude brighter, and maybe it deteriorates quickly. So you got to make it more stable. Maybe we want to make a different color because it's blue-green. Maybe we want red, yellow-red, or maybe we want whatever, right? We want a rainbow of colors. Okay. Many people worked on this. And now it's a very common technique to use these fluorescent proteins and put them in some cell types. So let's say you want to study, maybe you want to study dopamine cells, cells that make or release dopamine. You can put the protein for green fluorescent protein, the gene for green fluorescent protein in just those cells. So now they are different looking. They're literally green, and the other cells are just blank. So you can see them in a sea of cells. So that's a really useful and important tool. And you can do it in live animals because it is a gene. The cells make it. And so you can actually look into their brain and see the cell. You don't have to use like chemistry or chemical reagents to mark things. The cells make the protein. It is a gene. The cells make it. And so you can actually look into their brain and see the cell. You don't have to use like chemistry or chemical reagents to mark things. The cells make the protein.
How do they, if you put this in an animal and you want to look inside the brain, how do you look inside the brain to see if they're glowing?
There are different techniques. So we use a laser scanning technique. And this technique is also, there's so many interesting things. I know I keep saying that, but it is a laser scanning technique that uses a relatively long wavelength of light that can penetrate through relatively thick samples. So it's actually possible to point this laser through a skull that is partially intact, and you're just seeing the signal come back from the brain. So you are literally forming a movie or a physical picture of brain activity. And when I say movie, you can take pictures repeatedly and form a movie of what's happening over time. And this is really a valuable technique. It has some disadvantages, which I'm also happy to talk about, but it provides pictures. So you can see the same cells over time. So it's a really interesting possible tool for tracking the same cells over time to see how experience has changed the brain, because you're recording from the same ones. It's a powerful technique because of the specificity that I just told you about, because you can see the ones that are green, the cells you're studying. It makes it much easier to be repeatable. From animal to animal, you can say, I am studying the same group of cells. Whereas in the past, you simply couldn't do that. The tools were not there. And then the third reason the technique is valuable is actually kind of going back to the fluorescent protein. So if you wanted to follow up on that, I think this is a pretty interesting tangent to jump back to. So the fluorescent proteins, you see them. But what if we could make the signal means something, right? It's not just that it's there or not, it's that the signal, like the brightness of it, tells us about the internal state of the cell. And over the last, maybe since 97, 98, that's when they first were made. They basically would take green fluorescent protein and fuse it to different sensors. So let's say you wanted to study some internal molecule in the cell, calcium. I'm going to give an example. Calcium is an important molecule. It's related to the activity of the cell. Whenever there's calcium in the cell, the sensing element would cause the fluorescent protein to change. So you can use the brightness of the protein, the fluorescent protein, to track the activity of the cell. This idea has exploded in the last 10 years. You take the same principle, you take your reporter, your optical reporter, it's just a thing we see, you attach it to many different things. You want to sense glutamate, GABA, two molecules involved in making cells excitable or less excitable, serotonin, dopamine, acetylcholine, cannabinoids. There's now optical sensors of orexins. Many, many groups are developing these tools and making them more useful. And it's a way now of we could study all of these different molecules in the brain that we could not even study before because there was no way to selectively look at just this or that, right? We can, because the reporter is specific for, let's say, serotonin, all of the fluorescence is related to serotonin, right? Does that make sense? So it's like a real, that's a big deal in my opinion. These are like tools that are kind of some of the tools are a little bit rough, but this is going to be a game changer over the next 2 or 3 decades, like as they make them better, right? Just think like first iPhone, a little bit rough, right? And make it better and better and better. That's how neuroscience tools are. They're making them better and better and better until we reach some physics limitations.
Yeah, that makes sense because if you can parse, to use one of your words, if you can segment all the different cells and use that with color, then you can start to see connections, you can start to see movements, you could see regrowth, you can see death. And that's pretty fascinating because it's really getting more and more and more precise into what is happening inside the brain and those interactions. Yeah, that's really fascinating. And that goes back to just kind of the focus of your lab is to understand that the function of the brain and the function of the brain relative to the things that, we do in life, and how we react to, sensory input. That's really fascinating. I have it may be a weird question and maybe you don't study it, but do we know what causes hallucinations? Because, is that an outside thing? Is that an inside thing? Is that a protection thing? Do we know what causes hallucinations?
I don't, but there was a paper I saw recently where they gave psilocybin to people in an fMRI scanner and crazy things were happening. This is now my huge speculation, so you can even cut it out if it's crazy. But I mean, if like someone has like a minor epileptic fit and it causes triggering of certain neurons in your brain, maybe those neurons are embedded in a memory circuit, right? Or some memory, like I think about them as like constellations of cells. Like there's not a memory cell for like, right? This is now a controversy, but I don't think there's like a cell representing your grandmother. There's probably collections of cells that as a constellation or pattern encode that. And maybe through some unusual activation, it causes that pattern to be activated because you activate one by accident, it triggers a chain reaction. That doesn't seem crazy that could happen, right? It's just, why is it happening? And you can kind of come up with reasons for why it would have happened by accident, but it happens to be, if you think, a very difficult thing to study because how would you study it repeatably? You need a model that you believe is hallucinating, and then you need to study it. And so that makes it really tough. That brings you back to another reason why the brain is so difficult to study is that there are many phenomena that we really want to know about, but are nearly impossible to study in a real brain, which is the reason why we use other model organisms. I mean, the dream is to eventually do everything in like an organoid or something or computationally, but we're not there yet. Like if we were, you should be able to program a little robotic fruit fly to behave exactly like a fruit fly, or you should be able to like program a human, like a robotic human, to essentially. We're not quite there yet, but maybe we're getting closer. I don't know.
Yeah, I think we're a long way away, because even if those memories of your grandmother are brought up, is it which memory of your grandmother? There's so many memories. There's just millions of them. And so, yeah, it's super, super interesting. So how does the kind of the olfactory sensory, bulb there, is it affected with age? And how does that kind of, fit into the aging process? What do we know about that?
Great question. I have to say, I should have looked it up some more before coming here, but I'm not as familiar with how aging is really affecting the olfactory bulb itself. I mean, it's definitely true that people lose their olfactory abilities with age, and loss of olfactory function is a hallmark of some diseases, like people with Alzheimer's disease and Parkinson's exhibit decreased smell. Now, I think it's unclear whether it's true for many parts of the brain. And it just happens to be that maybe it's easier to see that or measure it, or maybe people haven't measured that there happens to be general declines in many things. So it could just be like some general dysfunction and it's not smell specific, although I would like it to be smell specific as a smell researcher, because that would be very interesting. But I'm not sure that is specifically happening in olfaction, but definitely there are deficits in those diseases. There is a colleague of mine who discovered that you do see deficits in kind of on a related point, where in rodents who are made obese by feeding them high-fat diets, you actually see deterioration of their olfactory systems, which is very, very interesting. It's still not clear whether this is due to like kind of a general inflammation or something specific to like signaling within the olfactory system. And that's, I think, one of the projects she's funded to work on. Give her a shout out, Dr. Deborah Westphal, who's one of my colleagues. Actually, she is a co-author on one of the papers we published from the Toffler funding. So one of my close colleagues. But very interesting phenomena. It really shows you that. I mean, I know I'm derailing from your aging question because but it really, going to the obesity question, so little is known about how different metabolic states affect the brain. I mean, it's also a very difficult question to ask because it's hard to do these experiments in a controlled and rigorous manner because so many things can change. But it's clear that will affect that obesity will dramatically affect the brain. And that is a very interesting problem because it's a major problem over the last couple of decades. And I think maybe even like 30 years ago or 40 years ago, everyone's like, oh, it's really a disease of willpower. But it's definitely a very complicated thing that has involved many functions and dysfunctions. Like I mentioned these cells that detect leptin earlier, which are a molecule released by your fat cells. And there are people who are born with leptin resistance, so they can't detect it and they have obesity problems. But curing that doesn't necessarily fix, well, it'll fix the problem in people who are leptin resistant, but it's not like giving too much of leptin causes or fixes obesity. There are many other signaling pathways that are involved in that process that are important. And I'll just give a very straightforward one. The stretching of your stomach is a cue used for knowing whether you're hungry or not. I mean, it's a mechanical stretch force. So somehow it's being transmitted to your brain, the stretch in your gut, right? It's just a mechanical signal. And that is somehow being integrated with all of the other chemical signaling processes occurring. It's a very, very interesting problem, which I think just not too long ago would have been thought of as something completely different.
Yeah, it's the more I listen to you, the more it's just, wow, we just don't know. We just don't know anything, do we? we just there's just there's just so much to think about, and things that I thought we knew. It's just we really don't know why that is. That's amazing. So
I tell my dad, I'm like, this is like a 500-year problem, maybe. Maybe I'm being not optimistic enough, but just the scale of it is very difficult. And we're going to have to create all sorts of new tools and technologies, like the imaging tool that we use in my lab. I mean, here's an interesting story. I think in maybe a PhD student in the 1920s predicted the phenomena that we use, but there wasn't the engineering to make it. And in the maybe 80s and 90s, a group at Cornell realized that now the technology existed that you could at least try to see if this phenomena occurred, which is called two-photon excitation. I'm going to leave it at that imaging technique. And it just shows you like there's this balancing that's needed of theoretical ideas. Engineering has to catch up to them. Biologists have to apply the tools to study it, realize what we need next, go back to engineering. Hopefully, engineering is using the theoretical background to approach new tools, which can then be used. There is this harmonious and symbiotic relationship between these two groups that is absolutely necessary. And now I've given you a couple examples: green fluorescent protein, imaging. These things go back decades, but it's only through this process that we're making progress. It's very interesting to see. And like, I've asked myself, maybe we should just wait for everything to catch up and then we'll just, solve it all overnight. But I think it's necessary to be trying to push us and to know what to do next. Like it's this iterative process that is what science is, and it's what's pushing our understanding of everything forward.
yeah, if you're not trying to understand what's inside the brain and how things are moving or interacting, then others won't try to create tools to get inside the brain to understand how it's working. And so, you need a problem to solve, and different groups, different different perspectives, different talents, experiences see those needs, and they go and they solve. And innovation is a very messy process. It's not linear, and it's not sure. You can't wait, because if you wait, you're not creating the need for others to solve that need.
I think that makes sense, right? It's definitely true in not just neuroscience, but I think in humanity and human discovery and advancing our civilization.
Yeah, I think that's right. So on that note, when you think about the future and you think about, just the next maybe 10, 15 years in your line of kind of focus here, what are you the most hopeful for? What do you see any kind of hopeful breakthroughs in understanding, or what do you see out there in the future?
It's probably so the ability to deal with a lot of information at orders of magnitude faster than we have is intimidating and a little bit scary, but also important and is a big deal. Like the amount of data that now neuroscientists can collect is often the bottleneck. Dealing with the data is the bottleneck, right? If you're making movies of brain activity, you have to deal with that data. And the tools and the ability to just kind of like, because of other engineers making the tools that we can grab onto and use and leverage, it just makes it much easier to do things that would have taken, maybe it would have taken 5 years to do something that takes like literally a week now. I mean, a couple decades ago. It really makes a big difference in the throughput. On the other hand, it does sometimes feel like an arms race, right? You're like faster, bigger, more, more, more. And yeah, it's because things got easier. So the standards are constantly changing of what's a reasonable thing to do as a scientist these days. I am hopeful, like, right, I also got into neuroscience. I was really interested in the brain. And I became more of a systems neuroscientist also because I could have gone in different directions. My wife is in the mental health field, and it just feels like there's not good ways of addressing a lot of diseases. So I'm so hopeful that through understanding the brain and like also through the process of working on problems, and even though maybe our questions are not exactly disease focused, it's that process of working on it helps people see more stuff and may lead to discoveries for treatments. I mean, I think it's fair to say that a lot of treatments for many disorders are not that different today than they were several decades ago. They're often like shotgun approaches that are putting increased amounts of this or decreased amounts of this in the whole brain. But I just told you the brain is many, many parts. And maybe there's dysfunction in only a small number of them. So we need ways of targeting treatments more selectively. And in a way, that's one of the things that we work on is like tools to target. We're understanding, but the same tools you might hope would be used to be more precise in a treatment. So, I mean, I'm very, very hopeful, but it is a very difficult problem, understanding different disorders.
Yeah. And I think you kind of hit it. Being a neuroscientist that has a systems focus, because it's not just the system of the brain, but you started the conversation with, that relationship to your gut or your immune system or to your nervous system or to, we could even spread it out even more to the environment which we live, the water, the food, the stress that we feel. Yeah, you need that kind of systems approach to even identify some of the problems that need to be solved.
Yeah, I thank you. You're going to use your argument in my next grant application.
So, well, this has been really fascinating. I think I told you before that the conversations that we have, few and far between, they leave kind of a visual understanding of what's happening when I go through my day-to-day life. Things happen and I go, yeah, we don't know why that happens. And, why is this and why am I reacting this way? Or why is my body signaling this? It's interesting that we don't know, but it's very hopeful that we have people like you that are trying to figure that out, because I do think that is the key. And you said it, understanding the function. And then we can get to like what happens when it doesn't work that way. And that is, how that will shine a light more so on how are these disease, manifesting. So, well, Doug, as we just kind of come to a close here, if somebody's interested in finding out kind of more what you do, or if they want to get in contact you to collaborate, or even maybe fund, how would they reach out to you? Is there kind of a contact information somewhere that you would direct them to?
There is. I have a website. It's Storace, STORACE lab.com. So storacelab.com. And there aren't that many people with my last name. So I will probably pop up at the top of my Florida State website as well. So I have a website through the Department of Biology and the FSU's program in neuroscience. So it's, but the storacelab.com will also take you to those links.
Great. Well, I really appreciate your time. I appreciate what you do and your focus and your dedication here. So thank you very much.
Thank you, Deborah.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.